Welcome. Today I'm going to be talking about phototransduction. Phototransduction is basically converting light into an electrical signal. Okay, so this starts off by light shining onto the photopigments of the photoreceptors. This is a rod cell. Okay, and rod cells, they have photopigments called rhodopsins. Okay, now the rod, because it's a rod cell, so rhodopsin. Okay, and rhodopsin consists of a retinal and opsins. So when light shines onto these photopigments, the retinal, which is in a state of 11 cis retinal, gets converted into 11 trans retinal. Okay, and this change will cause a conformational change in the opsin component. And this conformational change will activate transducin. Okay, and transducin is a G protein. When transducin gets activated, the GDP, okay, so let me just show you here, transducin. So initially is bound to GDP. When it gets activated, the GDP gets replaced by GTP. Then, as you can see here, the alpha subunit of transducin bound to GTP goes along and binds to phosphodiesterase. Phosphodiesterase is an enzyme that breaks down cGMP. Now, cGMP in the rod cells are normally bound to this cGMP gated sodium calcium channel. Okay, when cGMP is bound to this sodium calcium channel, there is an influx of sodium and calcium. Now when cGMP it gets broken down, cGMP levels inside the cell decreases and there are less bound to these channels. So then these channels get closed. Okay, and more of them close. Now, in this cell, which is not shown in this diagram, there are also potassium channels. Okay, now these channels allow the efflux of potassium. Now, the potassium channels are not affected by cGMP levels. Okay, they do not close or they do not open. So the potassium channels, the potassium ions, they keep moving out of the cell. In normal conditions, the cGMP gated sodium calcium channels are open as well as the potassium channels. Okay, now when they are open, the cell has a membrane potential of negative 40 millivolts. Now when cGMP is broken down, the cGMP gated sodium calcium channels get closed. So there are less positive ions going into the cell and there are more positive ions going out of the cell. So the inside of the cell becomes more negative. Okay, it becomes more negative, and the maximum negativity this rod cells can reach is around minus 65 millivolts. After that, any increase in light intensity will not have an effect, or will have minimum effect. So when the cell becomes more negative, okay, the cell becomes hyperpolarized. The fact that there are less calcium ions going into the rod cells, okay, this will cause less neurotransmitters at the end of the rod cell to be released. The neurotransmitters are glutamate. So there's less glutamate being released from the rod cell, okay, when it becomes hyperpolarized. And the reduced glutamate levels will stimulate the bipolar cells. The photoreceptor cell becomes hyperpolarized and the bipolar cell depolarizes. Alright, so in this diagram, you can see the rod cell with its discs. Okay, this is the rod cell. This is the bipolar cell. And this is the retinal ganglion cell. So when light strikes, okay, by the way, remember, light comes from this side. Okay, I don't know if you can see my, yeah, so light comes from this side. Okay, since remember, when light is striking the retina, the photoreceptors are actually at the back. Okay, they are at the back here. So when light reaches the photoreceptor cells hyperpolarize, and then they release less glutamate. Now this reduced glutamate levels will cause the bipolar cell to become more positive, okay, and depolarize. Bipolar cells have a graded potential, so when they depolarize, they will essentially release more neurotransmitters on this side. The ganglion cells, they will respond to the neurotransmitters released by the bipolar cells and cause an action potential. And then the action potential travels through the ganglion cell and to the optic nerve. Now it does get a bit more complicated, okay, because you do have on and off cells, on and off bipolar cells, but that's for another video. Okay, so in general, when light hits the photoreceptor cell hyperpolarizes and releases less glutamate. The reduced levels of glutamate neurotransmitters in the synapse between the photoreceptor cell and the bipolar cell will stimulate the bipolar cell to depolarize. When the bipolar cell depolarizes, it releases more neurotransmitters, which stimulates the ganglion cell. And then the ganglion cell will produce an action potential and send it to the optic nerve, which eventually goes to the brain for processing. Anyways, thank you guys for watching. That's it for today, and see you guys in the next video.